Aloha, welcome back to General Pharmacology. Let's move the PowerPoint slides and get started today. Today we're going to talk about cellular physiology, a little bit of cellular physiology. This is the second lecture of the introductory material. So let's go ahead and get started. All cells organize their metabolic machinery into small organizational packets called cells. I think you know this. And a long time ago, a guy named Robert Hooke looked under a microscope of a piece of cork and drew this. And this is the first impression that we know of that first shows cells under a microscope. Well, some organisms are just simple single cells, like prokaryotes are just simple single cells. Bacteria, simple single cells. Humans have billions and billions of cells, each specialized and very complex. And these regulatory systems that regulate these cells are specialized and complex. And so if humans were just simple single cells, pharmacology and physiology would probably be a lot easier. But humans, just like you, have billions of complex cells, and that's why it's your fault this is so difficult. It's not my fault. I'm not making this stuff up. All right, so here is a cartoon of a cell, a round cell, and a piece has been cut out of that cell so we can see the inside of it. And inside of a cell is a nucleus. Just like inside of an atom is a nucleus, the center of something that's different than the outside is its nucleus. And we'll use these terms throughout physiology. And so inside of the cell is a nucleus. And the nucleus is quite simply the cell's control center. And you can get on Wikipedia and look any of this stuff up. I know everyone tells you Wikipedia is horrible, and Wikipedia will be horrible when we start talking about drugs and diseases. But for basic science stuff, the first few sentences out of Wikipedia are really good information, and I've looked at it enough times, I think. And so if you want a much more in-depth view of any of this, you can start with Wikipedia, and then you're going to find even better websites after that. So the nucleus is the cell's control center. We could write giant books about it, but I think that's enough. All right. The plans for protein synthesis are in the nucleus. The plans for protein synthesis are coded in what's called DNA, and we're going to talk about that today. And there's some grammar lessons for you. Nucleus, nuclei, nuclear, not nuclear, nuclear. All right, so everyone knows what a nucleus is. It's the center of something that's different than the outside. Do not confuse that with nucleolus. Nucleolus is something within the nucleus where ribosomal RNA is stored. Ribosomes are proteins that manufacture proteins. They're made of proteins and some nucleic acid material mixed together if you want to make it complicated. But what I want you to know what I want you to know is right here. Ribosomes, well, that was the wrong direction. Ribosomes are proteins that manufacture proteins. And hopefully I say that 50 times before the semester is over, because we're going to start talking about antibiotics, and they're going to be called ribosomal inhibitors. And they interfere with the ribosomes in bacteria that make proteins. So ribosomes are proteins that manufacture proteins. Mitochondria are fascinating little creatures. They look like footprints. There they are flashing on my cartoon. They produce adenosine triphosphate for the cell using electron transport. Adenosine triphosphate is the energy currency. It's the main energy molecule for the cell. And so when the cell needs energy, in many reactions, in most reactions, it can use ATP as the energy driver for the cell. It'll get more complicated than that as we go along, but ATP is a great place to start. Mitochondria make ATP. What's incredibly interesting about mitochondria is they have their very own DNA. And the DNA inside of you, your mitochondrial DNA, came exclusively from your mother. Mitochondria are passed through generations through females. Men do not pass their mitochondria off to their offspring. Only women pass their mitochondria to their offspring. And so there's all sorts of interesting projects out there studying mitochondria and DNA and how far back we can look in human history tracing back DNA. If you read very carefully throughout your career, you will realize that the near entirety of the creatures on this planet all share a genetic code. Their genetic codes are very similar. Mitochondria are very weird. Seems like their genetic code came from outer space or something like that, which is interesting because mitochondria are thought to be individual organisms, their own little bacteria that invaded another bacteria and provided a benefit to it. That's what endosymbiotic. Symbiotic means they work together. Endo meaning inside of it. So an endosymbiotic bacteria. This little invader came in and entered a cell and provided it with energy. And I think that's an incredibly fun thing to think about when we think about history of life on this planet. And not only are mitochondria little invaders that invaded the first prokaryotes to provide them with energy, but mitochondria are essential in making acetylcholine, which is a neurotransmitter, which is essential in so many living creatures on this planet. So mitochondria would be a great course all by itself. Hopefully there's a mitochondria course out there I can take someday. All right, so this is a mitochondrion under an electron microscope. It looks like a foot, and these little places are cristae where they increase their surface area, and this is where electron transport and energy creation occurs, energy transfer occurs. And it looks like a little footprint, mitochondria. A little grammar lesson for those of you really interested in mitochondria. All right, endoplasmic reticulum and Golgi, and this cartoon is the same thing. When you take a real physiology class, there are differences, but for this is fine. Endoplasmic reticulum and Golgi, they're a network of pouches and tubes, and they store and transport the proteins around and remodify, repackage the cells. All right, so the bottom line is that when a protein is membrane bound, well, it's not made on the outside of the protein. It's made in the endoplasmic reticulum and then carried out to the outer cell membrane. So membrane-bound proteins are manufactured in the endoplasmic reticulum and then transported out. Somebody right there asked me this question in detail, and so that's actually when people came to the studio. All right, rough endoplasmic reticulum is called rough because of ribosomes attached to the endoplasmic reticulum. So if we look at the endoplasmic reticulum and we look at the little rough patches, those little rough patches are caused by ribosomes. And what are ribosomes? Ribosomes are proteins that manufacture proteins. So the ribosomes of rough endoplasmic reticulum are the ones that make the membrane-bound proteins. The ribosomes bound to the membrane, to a phospholipid bilayer, manufacture the protein within the bilayer, anchor it within the bilayer, and then that's transported out by the endoplasmic reticulum. Smooth endoplasmic reticulum is smooth because it doesn't have ribosomes. And so here's my electron microscope of rough endoplasmic reticulum. And let's make that a little bigger. Okay, and so see all these little spots? They're inside the endoplasmic reticulum. This is the extracellular space. See those clear places? That's the extracellular space. And then this is the endoplasmic reticulum. And you see these little spots? Those are ribosomes. And ribosomes are proteins that manufacture proteins. And ribosomes bound to the rough endoplasmic reticulum are busy making membrane-bound proteins. And there are lots and lots and lots of membrane-bound proteins. Hence, lots and lots of rough endoplasmic reticulum, which is fun to say if not study. All right, so now we're going to take a look at the cell membrane. That is the layer that goes around and encloses the cells. And here's my cartoon of a cell membrane. And the two things we see here are the phospholipid bilayer and these membrane-bound proteins. So the cell membrane encloses the cell and regulates the passage of substances into and out of the cell. And so there is a little flashing hairpin looking thing. There it is right there. We're going to take that and take a look at what that is. And what that is a phospholipid. Phospholipids are the building blocks of cell membranes. So phospholipids are the basic building block of cellular membranes, and they're made of a phosphate and a fat. And here we've just drawn the fat as a straight line, and we'll talk about why we do that. So phospholipids are the basic building blocks of all cell membranes. Cell membranes are made of a phospholipid bilayer. And not only are cell membranes made of this phospholipid bilayer, but that phospholipid bilayer also extends down to and is continuous with the endoplasmic reticulum and the Golgi. The nuclear membrane not only has a phospholipid bilayer, it has two of them. So you'll hear it called a diphospholipid bilayer. Anyway, the membrane around the cell nucleus has two phospholipid bilayers around it. All right, so you remember that inorganic phosphate that we talked about in the last lecture with those 3 negative charges. It is very hydrophilic, very water soluble. Phosphate is very water soluble and attracted to water. Fats, on the other hand, are nonpolar and they are hydrophobic. They do not like water, meaning lipophilic. They like fat. Hydrophobic and lipophilic mean the same thing. There's a few people out there that say there's a difference, and we're going to ignore them for this course. Lipophilic and hydrophobic are the same thing. Fat-loving, water-hating. Now, when we get into cellular physiology, it's not like chemistry. In chemistry, each of these lines is a bond between carbons. And then you get to cell physiology, and we're just too lazy to draw fat. So we just draw this zigzag line, meaning it's some kind of fat, because a fat's some kind of hydrocarbon. And maybe there's a double bond in there. There probably is a double bond in there. And so this isn't supposed to show any specific molecule. In cell phys, we just draw fats as these zigzag lines. So that's what that means, to make things even more confusing from the last lecture. All right, so if we take a fat and we put a carboxylic acid on the end of it, we take any fat and we put a carboxylic acid on the end, what do we have? We have a fatty acid. Like this is like some incredible ingredient in shampoo, and you know what that is. Here, all we've done is just taken a hydrocarbon, put a carboxylic acid on the end of it, and call it a fatty acid. All right, so here's my inorganic phosphate. They call it inorganic because there's no carbon involved. I think that's very silly. However, it sticks around. So that's not pi. That's inorganic phosphate, all right, with 3 negative charges. And here I have 2 fatty acids, and we've drawn any kind of fat with just some generic zigzag line. Please do not forget that there is a glycerol involved in the transaction. And so if we take a phosphate and attach it to 2 fatty acids, we have a phospholipid. I used to have a slide for this. Yeah. If we take, we'll go backwards a second. Instead of there's 3 places where this glycerol can react. 3-glycerol. Do you see where I'm getting with this? Instead of an inorganic phosphate, if we had a 3rd fatty acid right here and we put the glycerol in it, we'd have a triglyceride. Right? So triglycerides are very similar to phospholipids, except instead of having 3 fats, a phospholipid just has 2 fats and then the phosphate on the end of it. All right, so here we've made a phospholipid. And so on a quiz is the question, what is the recipe for a phospholipid? And everybody will put phosphate and lipids, and they'll all forget about the glycerol. All right, so this is why we draw phospholipids like that. The zigzag lines aren't supposed to denote anything other than a lazy way of drawing a fat. And as if that's not lazy enough, we'll just leave out the glycerol and draw it like that. And that's really a lot more work to draw than that right there. And so that's why these are the symbols for phospholipid, because drawing out the carbons is ridiculously complicated. All right, well, the amazing thing about a phospholipid, it's very similar to a detergent, is that one side is very water soluble and one side is very fat soluble. And so the phosphate end of the phospholipid is going to be very water soluble, which means hydrophilic. Fat insoluble, lipophobic, means the same thing. And so this is the end that's going to want to point towards the water. All right, well, the fatty end of the phospholipid is water insoluble, it's hydrophobic, and very fat soluble, lipophilic. And so, like if this was a detergent, this would be the part that would stick to the oily stain, and then that phosphate on the end would make it water soluble to be carried away. That's why we worry about soaps having lots of phosphate in them, because phosphates on phospholipids, phosphate is what we use to make these little fatty chains very water soluble. All right, you take a phospholipid, you put it in water, the phospholipid tails spontaneously will form a bilayer, especially in biologic systems. And so that's where the phospholipid bilayer comes from. If you put the phospholipids into water, those lipid chains will be attracted to each other, leaving the phosphate ends out pointing towards the water. And so our cellular membrane is made of a phospholipid bilayer. By convention, when I draw this phospholipid bilayer, what's above is always extracellular, what's below is always intracellular. All right, here we are back at the cell membrane. We've talked a little bit about the phospholipids. Cell membranes are made of a phospholipid bilayer. Because of the lipid center of the bilayer, most water-soluble substances will not pass through the membrane. Only teeny, teeny, tiny water-soluble substances like lithium can pass through the membrane, and that's even an interesting topic. And so in pharmacology, water-soluble drugs won't pass through the membrane because of that fatty center. And so now the cell can make the outside different than the inside, just like we do with the studio here. We like to keep the room very cool to keep the temperature perfect around all this equipment, and we don't want rain and salt getting on everything. It would destroy it. And so the way to maintain the electronics in the studio is to have a very carefully regulated environment, and we're surrounded by walls that allow us to do that. And your cell membrane does the same thing. It wants the inside to be able to conduct chemistry that's completely different than the outside. So the inside can be different than the outside. I think we've made that point. All right, let's talk about proteins. Here we have membrane-bound proteins. My white things there flashing are membrane-bound proteins. Proteins are made of chains of amino acids, very long chains of amino acids, and proteins are produced by ribosomes. Ribosomes are just quite simply proteins that manufacture proteins, and proteins have every function, every mechanical function in the body is mediated by a protein. Pretty much every function in the body is mediated by proteins. They are the little building blocks of our cell machines, and the proteins are made of amino acids. So when we go through the books, when we go through the medical books and the pharmacology books and the physiology books, anytime you hear the word globulin or globin, as in glob or globe, we're talking about a protein. So an immunoglobulin is a protein that's made by the immune system to fight with infectious foreign substances. Hemoglobin is the stuff that makes your blood red when it's oxygenated and blue when it's not. That's hemoglobin. See that globin at the end? That is a protein. Anytime something ends in the word ASE, ACE, that is a protein. Like a protease is a protein that modifies other proteins. Transcriptase is a protein that transcribes. Kinase, kinase from kinetic. That means that's a protein that's involved with moving a phosphate onto or off of another molecule. So when we talk about receptors, when we talk about enzymes, and even when we're talking about ribosomes, we're talking about proteins. We'll talk about free proteins that are made by free ribosomes, but we're also going to spend a lot of time talking about membrane-bound proteins. And again, those are made by ribosomes in the rough endoplasmic reticulum. So again, by convention, I'll use this cartoon a lot in the pharmacology class. This is a protein. That's my phospholipid bilayer. This is the extracellular space outside of the cell, and this is the intracellular space inside of the cell, each and every time. And we will talk about free proteins in pharmacology, but when we use this diagram right here, we will be talking mostly about membrane-bound proteins. For instance, the sodium-potassium pump. We're going to talk about some proteins that we're going to talk about all semester long, and we're not going to dive too deeply into this. What I want you to understand is some of these words that we're going to use, they're just quite simply proteins, like the sodium-potassium pump is a great protein to talk about. It uses ATP for energy. Is my ATP step in there? No. It uses ATP to convert the membrane into a battery, into a capacitor. That's what the sodium-potassium pump does. It converts ATP energy into ionic electrical energy across the membrane. And it's just quite simply a protein. And something important I wanted you to see is that the little gears are shifting back and forth between one position and the other. When a protein carries out a mechanical function, it just shifts back and forth between one shape to another shape and back and forth and back and forth, and it's able to ratchet or pump these ions in both directions. Here is something called a ligand-gated ion channel. To start out, again, everything above the phospholipid bilayer is extracellular. Everything below the phospholipid bilayer is intracellular. And that sodium-potassium pump has created the sodium force. Now sodium wants to get into the cell, but sodium can't just spontaneously pass through the membrane. For sodium to get into the cell, it needs some kind of facilitation, some kind of transport. And here is a ligand-gated ion channel. All right, so the ligand is some kind of chemical signal. Maybe it's a neurotransmitter, some kind of hormone from the body. Maybe it's a drug from outside of the body, an exogenous drug. So that's what the ligand is. The gate means that once that chemical signal hits that receptor there, it's going to activate the protein and something's going to happen. So my proteins are gray when they're inactive, and then my protein cartoons turn white when they are activated. All right, and so it's a channel, the channel through the membrane, so this ion can pass, an ion, ligand-gated ion channel. So here my chemical signal comes in contact with the receptor and activates the receptor so that sodium can come rushing in. We'll talk about this during excitable membranes, like when we talk about neurotransmission, when we talk about cardiac conduction. We'll talk about these systems again. Well, here's a chemical signal that's activating an enzyme. What the enzyme's doing is causing a chemical reaction inside of the cell. Look, this is just something that opens a tube that allows the sodium to come rushing in. Here, a chemical signal on one side of the cell is causing a chemical reaction to occur inside of the cell. And these are the sorts of things that we're going to talk about when we talk about pharmacology class. Opening ion channels, chemical signals that cause chemical reactions to occur inside of the cell. And these are all just quite simply proteins. Here a chemical signal is activating a chain of proteins, and here we're ending up with adenylyl cyclase. Adenylyl cyclase converts ATP to cyclic AMP and creates this second messenger system for us. We're going to talk about that neurotransmission. But what I wanted you to see is just more complicated than, oh, well, here the chemical signal has come in contact with an enzyme, and that enzyme starts having a chemical reaction. That's pretty simple. Most of the signaling chains have these little signaling proteins in between. Here we have a G-coupling protein, and so if you read about adenylyl cyclase, they might talk about coupling proteins. And so we'll try to keep our coupling proteins at a minimum around here, close to 0. But when we talk about cancer and chemotherapy, keep in mind this chemical signal that's going on here that's affecting the nucleus, like on my side of the room. There will be 20, 40, 50 coupling signaling proteins from one end to the other. And so when we start talking about cancer chemotherapeutic agents, we'll see that chemical signals will affect all sorts of signal chaining down to the end result. And so that's why I wanted to show you all that. This is just quite simply how the body uses proteins, all sorts of different ways. All right, adenylyl cyclase converts ATP into a second messenger called cyclic AMP, and this is something that we'll start talking about again when we get to neurotransmission. All right, well, to get glucose into the cell, we need to facilitate that transport. So here, again, is my protein, and that protein is allowing glucose into the cell because glucose is too large to pass through the phospholipid bilayer without some kind of facilitated transport by the protein. And here, that sodium force that we created with the sodium-potassium pump is driving glucose into the cell. And again, my little gear should turn back and forth instead of turning in circles like that. Same thing with amino acids. The way we transport amino acids into the cells is very similar to the way we transport glucose into the cells. Glucose is for energy. Amino acids are for building blocks of proteins and all sorts of other stuff. But you'll notice as we go through that amino acids and peptides and glucose, they have a lot of similarities. We can take amino acids and make glucose. So just keep that in mind. This facilitated transport is required. Protein facilitated transport is required to get not only glucose in the cell, but the amino acids into the cell as well. All right, well, somewhere in neurotransmission, we start talking about acetylcholinesterase, which is a protein enzyme that breaks down acetylcholine. It divides acetylcholine into choline and acetate. And if you recall from your extensive and thorough understanding of physiology, that acetylcholine is in the neuromuscular junction. We're going to talk about other places that we're going to find acetylcholine. But to make the acetylcholine signal stop, something has to make it stop. And one of the strategies that the body uses to make neurotransmitters stop their signal is to just quite simply break them down. And so acetylcholinesterase breaks down acetylcholine. Well, it makes sense then, if I block the acetylcholinesterase, if I block this step right here, I'm going to end up with more acetylcholine. And that's a point that goes over everyone's heads, usually the first time I cover it. And so I thought we'd cover it now under proteins, because acetylcholine esterase is a protein that divides acetylcholine up into its parts, acetate and choline. All right, proteins are made of these long chains of amino acids. We'll take little amino acids and make oligopeptides. We can take single amino acids, make neurotransmitters. However, big proteins are made of very long chains of amino acids. And so if you frequent Wikipedia, you might come across this excellent description of amino acid. Here's my amino group. There's the carboxylic acid group. R is just a side chain or variable group. The alpha carbon always has a hydrogen attached to it. Not always, but here our alpha carbon has a hydrogen attached to it. And then how these things are arranged around that alpha carbon, that center alpha carbon, determine whether it is left-handed or right-handed version of the amino acid. Now, your two hands are identical to each other, and yet they are exactly the opposite. So be careful about words. We're going to start talking about medical words here very soon. But we'll talk about left-handed and right-handed versions of amino acids. And that left and right-handed version just quite simply has to do with how they're arranged around the alpha carbon. All right, well, if we take these amino acids and we put them into chains, notice the carboxylic acid group of 1 is lined up with the nitrogen, the amino group of the next group, of the next amino acid. And what I wanted to point out is those little water molecules that are going to disappear when the reaction occurs. And so when a peptide bond occurs, the water molecule is removed from it. And so when we hear about breaking a peptide bond, when we hear about something that separates a peptide bond, the way it does it is to hydrolyze it. Take that water molecule and stick it back in the bond so they can be split apart. Right? And so if you hear about hydrolyzing, that's because that water is taken out during the peptide bond step. And then when we use things that are like a protease that break the peptide bonds, they just quite simply insert the water back into the chain to break it up. All right, so now we have these peptide bonds. We can start making chains of amino acids. right, so amino acids are also called peptides. If you put several peptides together, you'll have polypeptides. Oligo means little or short, and so oligopeptides are short chains of amino acids. And so we'll talk about insulin and glucagon and vasopressin, the same thing as antidiuretic hormone. They are quite simply polypeptide hormones. We could consider them oligopeptide hormones. And so small amino acid chains can make these hormones and drugs, but it's very long amino acid chains that actually form proteins. All right, so here's our generic structure of all amino acids. Again, there's a side chain or a variable group. There's an acid group, an acid moiety, and then an amino moiety as well. And the amazing thing about amino acids, it's that R group, that side chain that gives the amino acids their incredible property. The amino group and the acid group is just quite simply how we connect the box cars together on the train, and what's important is what's in the box. And so what's in the box is this R group, and then how we connect them is through amino groups connecting to the acid groups. All right. I don't expect you to memorize this unless you're functioning at senior level biochemistry. When I took biochemistry more than once in my career, we were expected to memorize these. And you're welcome to do that, but what I want you to see if you're early in your career is how the R group changes from one amino acid to the other. And we just take 21 of these, and every machine in our body, every process that occurs with a protein, is just quite simply made of these few building blocks. I mean, it takes us more letters of the alphabet just to make words than it does the body and its collection of amino acids and put them together and make all this miraculous machinery, which is just incredibly fascinating to me. All right, so here's glycine. Glycine does not have a left or right-handed version because that alpha carbon has hydrogens on both sides. So glycine is the simplest amino acid. It does not come in a left and a right-handed form. However, your body does use mostly the left-handed version of amino acids, and so what they're trying to show here, this is not a hydrogen bond. That just means the hydrogen is pointing away from the screen. And then this right here is supposed to show a methyl group. There's a carbon right there, and so that methyl group, that R group, is pointing towards us. That's what that convention is supposed to show. And I don't need you to know any of that unless you're trying to learn the difference between left and right-handed versions, and then maybe this will be helpful for you, this convention. All right, so L-alanine, what is its R group? It's just quite simply a methyl group right there, a carbon. Well, L-arginine, its R group is very large and complex, and when we look at complex ring structures with nitrogens in them, we might see that right there. And so these R groups and the proteins will come together and form these structures that do really incredible things. If you read about the history of asparagine, somebody went through a lot of asparagus to come up with asparagine. And notice this group on the end, the amide group on the end of this ketone. And so this is going to be very water soluble, very complex R group on the asparagine. Aspartic acid has a carboxylic acid on the end of its R group. Cysteine has a sulfur group, and you'll hear about sulfur bridging. If you look at pictures of immunoglobulins, the light chains and the heavy chains are put together by sulfur bridging. If we look at a picture of the hemoglobin, the 4 subunits of hemoglobin are put together with sulfur bridging. So when we see sulfur bridging, cysteine is a very likely candidate because we can take that hydrogen away and then make that second covalent bond. Glutamic acid quite simply has carboxylic acid on the end of its R group. Glutamine, again, has this complex structure on the end of its R group. Histidine has this complex ring structure on the end of its R group. Histidine is what we'll use to make histidine is what we use to make histamine, hence the use of the medications antihistamines. Isoleucine has this complex R group on the and notice this isoleucine These are hydrocarbon chains. R group is a hydrocarbon chain. So it's going to be fat soluble, and so isoleucine might be something that the isoleucine would be something the body uses to anchor the protein into the phospholipid bilayer. Same thing with leucine. It has a hydrocarbon as an R group. Lysine has an amino group on the end of its R group. Methionine has a methyl group right here, and so bridging with cysteine just occurs by removing the hydrogen. Sulfur bridging with methionine involves demethylation. Hence the word methionine, methyl group. Phenylalanine is how we're going to start making dopamine. Everybody else will tell you that we start with tyrosine, but if you start with phenylalanine and put an oxygen group on it, you'll have tyrosine. If you put another oxygen group on it, another hydroxy group on it, you'll have a dioxy phenylalanine, and that's what the word dopa comes from. And so we'll make dopamine starting with phenylalanine. Phenylalanine is essential amino acid. You can't make this, you have to eat that. And then we can turn this into tyrosine. So I want you to know the phenylalanine to tyrosine step. And when we see tyrosine, there'll be a little hydroxide group right there. Proline's very interesting because its R group comes back and reacts as part of its nitrogen group. And so when we see amino acid chains that take a quick change in the direction, when we're going one direction and then we make a hairpin turn and we go the other direction, we know that proline is involved. And we'll see that again. We're going to talk about these things called serine proteases. And when we talk about a serine protease, the knife edge of the protease is that oxygen group right there. And we'll see that again when we talk about serine proteases. But there's a little hydroxide, a little oxygen group on the end of its R group. Threonine has a complex hydrocarbon chain with a hydroxide group on it. Tryptophan is what we're going to use to make serotonin, 5-hydroxytryptamine. By removing that carboxylic acid right there, we'll have a tryptamine, and it starts with L-tryptophan. Tyrosine, remember phenylalanine? We started with phenylalanine, we stuck a hydroxide group on it. Now we have tyrosine. So we can manufacture tyrosine, but we need phenylalanine to do that. Valine has a smaller hydrocarbon branched structure on the end of its R group. All right, well, that's not the point. The point is this, the point is this right here. It's the property of the side chains that give complexity to the many proteins we're going to discuss over and over again. I don't need you to memorize these. I just want you to know that the body is able to manufacture all of the incredible machinery that it does just by using these simple amino acids right there. And I just want you to see that just these R groups, these different R groups, this 20 or so R groups is all we need to make all of the complex and incredible things that go on with proteins in the body. All right, so again, we're going to talk about acetylcholinesterase because by the time I get to that neurotransmission, I don't think people really get it. And so we'll talk about it again. Acetylcholinesterase breaks down acetylcholine. All right. Acetylcholinesterase is kind of like the drain that makes acetylcholine go away. So if we block that drain, we end up with more acetylcholine. All right. And we'll talk about that as we go through pharmacology. But acetylcholinesterase, again, is a protein that breaks down the neurotransmitter acetylcholine. So acetylcholinesterase hydrolyzes acetylcholine into choline and acetate. All right, so we won't call acetylcholine one of our amino acid-based neurotransmitters, but acetylcholine is still very close to neurotransmitter-based, amino acid-based neurotransmitters, and it's still broken in half with hydrolysis, just like any peptide bond. So there's my drawing of acetylcholine. Notice my nitrogen's making 4 covalent bonds, so it's positively charged. And we're going to break it right there and divide it into choline and acetate. Acetate, acetic acid, vinegar. And I showed you this picture. Acetylcholinesterase is a membrane-bound protein that takes acetylcholine and breaks it down into acetate and choline. All right. Now, when we take the acetylcholine and we split it into choline and acetate, see that little orange oxygen right there? And remember, oxygen makes two covalent bonds, so there's a hydrogen missing right there. All right. And then there's a hydrogen missing right there. So to put these two together, we need a water group. We need hydrolysis to split those. And what I want you to see is where this oxygen comes from, right here. All right, so acetylcholinesterase is a serine protease. That means the amino acid serine is involved. And right there is the knife edge. That little oxygen group is the knife edge of the protease. And so the protein is going to lay the molecule perfectly down across this knife edge so it can split the peptide bond apart. Here it's the bond between the acetate and the choline. All right, so here's my acetylcholine again, and there's my acetylcholinesterase. That's not to scale. If this was actually acetylcholine, then acetylcholinesterase would probably be about the size of this building, somewhere between the size of this room and this building, all right? Because again, acetylcholinesterase is made of these long, long, long chains of amino acids, and acetylcholine is about the size of an amino acid, all right? So here's my acetylcholine. Here's my acetylcholinesterase sitting in the membrane-bound protein. And in this long chain is the amino acid serine putting its hydroxide group up into the active reactive group like a knife edge. All right, and then here I have a little bowl. Oh, there's my serine. It's the reactive part. Okay, my little knife edge. All right, well, when these chains come together, one of the things is these chains that have the amino acids that have the little aromatic rings on the end of them, like phenylalanine and tyrosine, they come together and they form this bowl with this overall negative charge. And so this bowl, which sits in this place, has this negative charge to it. And what's it going to do? It's going to attract the positive charge of the acetylcholine. All right. So now the acetylcholine's been attracted to that negative charge, and it sits perfectly in place to where it lays this oxygen across the serine, and the serine can react and break apart the acetylcholine. And this is how we get the signal to stop. When acetylcholine goes into neuromuscular junction, well, the signal would just go on forever, almost, until it's stopped. And so to have quick on, quick off with our muscles, we release acetylcholine, and then the acetylcholinesterase just breaks it down immediately. So we have to keep releasing neurotransmitter to keep the signal going because it's the acetylcholinesterase that makes the signal stop by breaking down the acetylcholine into choline and acetate. Well, then a water molecule is able to react with, since the serine lost its oxygen group, the water molecule is there to replace that oxygen, basically resetting the acetylcholinesterase. And so these enzymes function like, the enzymes function in a way that they are not consumed in the reaction. They function like a catalyst. A catalyst facilitates a reaction, but is not consumed in the reaction. And so here we can see that the serine was consumed in this reaction, but then resupplied. And so this reaction can occur over and over and over again with the same protein. The protein's charged. It has a negative charge in this ball to attract the nitrogen, and then as soon as it's broken apart, there's a conformational shift, a change in the shape of the protein that causes this bowl to become slightly positively charged and push that quaternary nitrogen away. And this can happen over and over and over again. A single acetylcholinesterase protein can break down 25,000 molecules of acetylcholine every second, and that's just one acetylcholinesterase. So we'll talk about acetylcholinesterase inhibitors as we go along. However, I wanted you to see what a serine protease was, because sometimes that word will come up and they'll say, oh, well, this is a serine protease. Any kind of protease is going to work just like this. the fact that serine is involved in the knife edge. They'll call it a serine protease. And I wanted you to see where that comes from. Let's talk about ribosomes. Ribosomes are proteins that manufacture proteins. Ribosomes use single amino acids and will form peptide chains. Remember, rough endoplasmic reticulum is rough because of ribosomes. So again, ribosomes are proteins that manufacture proteins and using messenger RNA, mRNA, as the plans. So mRNA is copied from the DNA in the nucleus and then moved out to the ribosome. And so here is a picture of some kind of cell, and we're going to expand that. Oh, we remember this picture. And remember that rough endoplasmic reticulum with all those little spots? Those little spots are ribosomes. And when we get to antibiotics, they're going to talk about the big part of the ribosome, the 50S subunit inhibitor, the 50S subunit. And then we'll talk about the little parts of the ribosome, the 30S subunit. And so I don't need you to know the difference between 50S and 30S. I just want you to realize that there is a difference between the two subunits. All right, ribosomes form chains of amino acids, and so that's what we see occurring here. Each color is a different amino acid, and this just goes on and on and on until the protein is formed. Well, it's quite simply the sequence of amino acids that determines the properties. And so one of the properties is that the amino acids will come together in such a way that they form springs. Alpha helices, alpha helix is a spring. And how do these springs occur? Well, you take certain amino acids, you put them together in chains, they spontaneously form springs. Right? And so alpha helices are very common in protein structure. Not only as springs store mechanical energy and then release mechanical energy. Here pretty soon we're going to start thinking about pharmacology as locks and keys. And if you think about a key going into a lock, there's all sorts of springs involved, and it's the same thing. However, something else these springs are good for is sticking their R groups, their fat-soluble R groups, into the membrane-bound protein so that the protein is solidly anchored in the membrane. So when you see these little curly-Q ribbons on a drawing of a protein, those are just quite simply amino acid chains that have formed an alpha helix. If you go to Wikipedia and find hemoglobin, this might still be there. It was there 14 years ago. It's probably still there now. And here are the 4 subunits of hemoglobin. If you're into advanced things like beta thalassemias and alpha thalassemias, those have to do with disorders of the protein structures of hemoglobin. And these are 4 separate proteins that are put together by sulfur bridging. Inside of the reactive part of the hemoglobin is the reactive group, the coenzyme where iron is required. And the iron will go from its red state to its blue state, depending on whether it's connected to oxygen or not, just like rust. You've seen, if you're familiar with any kind of blue metal, you know if you leave it out in the rain and the oxygen or the salt water and the air, it'll turn into red metal. And turning metal from blue to red is the same thing that happens when your blood turns from blue to red and back and forth, whether these reactive groups are connected to the oxygen. The micronutrients that are required to drive these coenzyme systems are things that those of you who are studying nutrition are going to want to focus on, but these are very important to health and that sort of thing. And we're not going to talk a lot about that in pharmacology, but the micronutrients are essential for those reactive groups to occur. Back to our regularly scheduled program, something that I want you to see is that changing shape from one shape to another is how the hemoglobin or any protein functions. And so here hemoglobin is going from its oxy to its deoxy state, quite simply by shifting position. And notice the springs store energy and release energy, so the protein can go back from one shape to another. And that's called its conformational shape. That's called its quaternary structure. All right, so rather than put the amino acids together in a sequence that forms a chain, we can put the amino acids together in such a way that they form something called beta pleated sheets, like corrugated roofing. Corrugated roofing, very stiff, They stack together very nicely, so they can't be pulled apart. And so when we want stiff structures, your body makes beta-pleated sheets. And so we'll see these in ligaments, tendons, bones, stuff that we don't want to be real stretchy. We'll see a lot of beta-pleated sheets. And so those are for very stiff structures. And that's just quite simply determined by the sequence of amino acids that the body uses when they put the chain together. The amino acids will just spontaneously form these sheets. The sheets will be drawn with these arrows, and I don't need you to know the difference between parallel and anti-parallel, but I will point out that on a beta-pleated sheet, when you're going from one direction, then you take a hairpin turn and go the other direction, that proline is an essential step. And so you might hear muscle building formula stuff be rich in protein because muscles need beta-pleated sheets, and anti-parallel beta-pleated sheets need protein to change the direction. I think they leave that out of the commercials. All right, so then if you're Googling around the internet looking for pictures of proteins like I do, you might come across this, where we have little alpha helices that form springs, and then these little beta-pleated sheets that form stiff structures. And here pretty soon we're going to talk about locks and keys. And if you think about the inside of a lock, it has a bunch of stiff structures and then a bunch of springs. It's the same thing with this. We have stiff structures and then we have springy structures. And again, we have a reactive group that require the micronutrients to drive that reactive group. Those are called coenzymes. And if you're in the nutrition business, you're going to get to read all about that. All right, so the primary structure is just the chain of amino acids. What order do the amino acids go together? And that primary structure determines what? Whether alpha helices form or beta-pleated sheets form. That'll be called secondary structure. All right, well, the overall shape that it takes, its glob shape, is called its tertiary structure. And then when it shifts back and forth from one form to the other so it can perform its functions, that's called quaternary structure. And all of that is just quite simply determined by the sequence of amino acids. 20 amino acids, 21 amino acids, all we need to drive all of the proteins. I think it's incredible, which is why I spend all my time trying to study stuff like this. So where do ribosomes get their information to put this stuff together? All right, well, it's stored, the genetic code is stored in our DNA, and the DNA is translated in messenger RNA, carried out to the ribosome. All right, so here's my messenger RNA down at the bottom, and the ribosome is reading this messenger RNA, forming this amino acid chain up here. And then messenger RNA comes from the DNA in the nucleus. All right, so let's talk about the nucleic acids, RNA and DNA. RNA and DNA, our nucleic acids, are formed of chains of nucleotides. And they both use the same nucleotides. RNA uses GCA and U, and DNA uses exactly the same for nucleotides, GCA and T. There's a little teeny tiny difference between T and U, hence the different names. But I reject out of hand anyone who says there are 5 nucleotides. There are really just 4, and T and U change a little bit depending on whether we're talking about RNA or DNA. All right, it's like a bicycle with training wheels. It's not a different bicycle. It's the same bicycle, just has a little training wheel on it. All right, and so we want DNA to spin and be real tight. And we're going to take one of the training wheels off of the U so we can tie the DNA up really tightly. All right, so when I draw these on my cartoons, they're going to look like this. DNA and RNA use sequence of nucleotides to code for the sequence of amino acids. And any 3 nucleotides, the 3 nucleotides that code for a single amino acid, are called a codon. And so a codon is just quite simply the message for a single amino acid. Now, the codon is specific for an amino acid. That codon right there can't be for any amino acid. It can only be for one amino acid. And when I was making these slides up, I thought, well, gee, I wonder what the codon is for CAC. So I had to Google that. I'm sure I got it off of Wikipedia. Oh, yeah, the codon for histidine is CAC. Now, there might be other codons that code for the same amino acid, but that codon right there can't code for anything else. Any codon cannot code for anything else except one amino acid. And so CAC is the codon for histidine, whatever I was reading at the time. Not that I memorized stuff like that. Well, notice that the CAC of the codon matches perfectly with the GUG of a transfer RNA that's carrying an amino acid. It puts it in the perfect place, and once that peptide bond occurs, it causes a conformational change, pushes it away, and gets ready for the next step. So we can repeat the process. And then the codon for GGA is going to attract transfer RNA, putting the perfect amino acid into place. Oh, there's the perfect code for GGA. Is CCU, because these are going to match perfectly like little puzzle pieces spontaneously. They don't have to go look for it. It's just like, oh, there it is, stick. And puts the amino acid right in the perfect place. Once the amino acid has made its peptide bond, the transfer RNA is sent away. And that's how this occurs. Right? Made ridiculously simple. RNA stands for ribonucleic acid. DNA stands for deoxyribonucleic acid. Right? So you should know these terms. Right? And again, they're nucleotides. And their nucleotides are made of a nucleobase, a ribosugar, or the deoxyribosugar, and then a phosphate. When we talk about cancer and chemotherapy, we'll talk about purine antagonists. And what we're doing is trying to interfere with the formation of DNA and RNA in the cancer cell. And so we might use purine antagonists. If you read carefully, you might read about pyrimidine antagonists. I don't need you to know the difference between the two, but I want you to know that these are words that we use to describe parts of the nucleotide. All right, so these are the purines. Adenosine and guanine are used in DNA and RNA. Adenine and guanine are the purines used in DNA and RNA. Cytosine is used in DNA and RNA. Uracil and thymine only differ by that little methyl group right there, that little training wheel. The training wheel is on the DNA. I think that's correct. The training wheel is on the DNA so it can wrap more tightly, not on the RNA. All right, so that's why U and T are basically the same thing except that little training wheel right there. Hence the four nucleobases. Adenine, guanine, cytosine, and then uracil and thymine are the same thing except that little thing right there that comes on and off depending on what we want. All right. I see this a lot, hence the slide. Maybe this is in your notes. Thymine is a pyrimidine nucleobase. A thiamine is vitamin B1. Please do not confuse these terms. They sound the same coming out of your mouth. They are two different things. Okay. Well, that's all complicated to draw. I'm going to draw my four nucleobases just like this, AGC, and then U or T, depending on if we're talking about DNA and RNA. All right, so let's start with ribose, as in ribosome, as in ribosugar making nucleic acids. That's ribose. If we take that oxygen away, I think we have a deoxyribose. That's where that comes from. You know, RNA, DNA, okay? Deoxy. All right, so if we put an adenine, if we put the nuclear base adenine on our ribosugar, we have adenosine. The same adenosine we're going to use in supraventricular tachycardia, paroxysmal supraventricular tachycardia. The same adenosine we're going to use to make ATP. Right? And so we've taken a ribosugar and attached an adenine, and now we have adenosine. If we take a ribosugar and attach a guanine, we have a guanosine. If we have a ribosugar and attach a cytosine, we have cytidine. If we attach a ribosugar to the nucleobase uracil, we have a uridine. And if we attach it to a thymine, we have a thymidine. And so, again, the one that I wanted to show you here is where all these words come from. Adenine, adenosine, the only difference is the the ribosugar. Guanine, guanosine, just the ribosugar. Cytosine to cytidine. Uracil becoming uridine is just quite simply that ribosugar. Thymine becoming thymidine. All right. So now we've talked about where a nucleoside comes from. A nucleoside is a ribosugar and its nuclear base. If we put a big orange phosphate on it, then we have nucleotide. Tide, the detergent, and a yellow box, which is our color for phosphate. Tide's full of phosphate because detergents are very similar to phospholipids. All right, so what's the difference between a Zide and a Tide? Tide has phosphate in it. Okay, Zides don't. All right, so we went from a monophosphate nucleotide to a diphosphate nucleotide. If the nuclear base was A, this would be ADP. If it's G, it'd be GDP. If it was C, it'd be CDP. If it was U, it'd be UDP. If it was T, it'd be TDP. You'll see ADP all the time, but not GDP maybe, but the other ones you don't see so much in the books. And then the same thing with a nucleotide triphosphate. If the adenine is the nuclear base, this would be adenosine triphosphate. If the guanine was the nuclear base, it would be guanosine triphosphate. You'll see ATP and GTP, but whether you see CTP and UTP and TTP, that's a little bit more rare. All right, so, but I wanted you to see where this nomenclature came from, where these words come from. They use the little curly Q to denote that third phosphate because it is hanging on by a thread. It's like this hair trigger, and it is ready to go boom, releasing an enormous amount of energy. All right. So the same ATP that we're going to use as the energy currency for the cell is the same building block we're going to use for genetic material. So be careful. When I was in college, I wanted everything to just have one purpose. I didn't like things multitasking, and I can tell you right now, the body does it all the time. It's like a brick. What can you do with a brick? Well, you build a sidewalk. Well, is that all the brick does? Yeah. No. What can you do with a brick? You can build a building with it. You can build a barbecue grill with it. You can make a doorstop out of it. And there's just a lot of things to do with your bricks. And your body does this all the time. So don't get in the habit of thinking this brick is only for making sidewalks. It's not the case at all. Your body is going to use ATP to make genetic material. Your body's going to use ATP as a second messenger system. Your body's going to use ATP as the energy currency for the cell. All right, so you take these nucleotides and you put them together, they form chains. All right, so now we have one end of the chain that points to the nucleobases, and then on the other end of the chain, the other side of the chain, are these phosphate groups that are attached to each other. All right, so that's all complicated to draw, so I'm just going to draw them like that. All right. And then, again, we're talking about making, drawing pictures of nucleic acid. So I've made my chains into nucleic acids, and you're going to notice that my cartoon pieces look like puzzle pieces. Why? Because A is only going to match with U or T, and C is only going to match with G spontaneously through hydrogen bonding. So nucleobases only match in specific pairs that allows them to spontaneously match, that a robotic arm doesn't have to go find it and stick it in. The base pairs will spontaneously match with each other. So G will only bind with C, and that's through triple bonding. A only binds with T. Through a hydrogen bonding. I'm sorry, let's go back. G only binds with C in both DNA and RNA, and that is through 3 sets of hydrogen bonds. A only binds with T through 2 sets of hydrogen bonds, or U if we're talking about RNA. All right, so there's a single-stranded nucleic acid. There's a double-stranded nucleic acid. All right. So DNA is a double-stranded nucleic acid, and you'll see the directions run the opposite direction. Here the chain is formed in one direction, and then here the chain is formed at the other direction. So the 3 and the 5 prime refer to the ribose sugar. And so the 3' is the hydroxide end, the phosphate looks like it's the 5' end. I don't expect you to memorize that. I just want you to know that they run in opposite directions, and that G only binds with C, A only binds with T, or A only binds with U, depending on whether we're dealing with DNA or RNA. And if you look at cartoons of DNA, double-stranded DNA, you'll see it It wraps in this helix, and you might see that there's a tiny groove, a smaller groove, and a major groove. And that's actually important when you get at senior level biochemistry stuff. I copied this animation somewhere because it was cool, because we see this. This is where that double helix shape comes from. DNA is a double-stranded nucleic acid, and it looks like a ladder. Looks like the spiral staircase, and I want you to see that the steps of the ladder, the steps of the staircase, are the nucleobases spontaneously attached to each other, and then the rungs, the sides, the rails of the ladder are the ribose sugars in their chains. And so that's what that is supposed to be showing you. All right, so DNA is a double-stranded nucleic acid. The base pairs are stuck together very tightly, and the DNA is wound up in a little teeny tiny a little tiny spiral wrapped around all sorts of regulatory proteins very tight. Well, if we want to read the DNA, what do we have to do? We have to unwind it and unzip it and open it and transcribe it. And so there's all sorts of regulatory proteins involved in unwinding and unzipping and transcribing that DNA. And so here we might talk about helicase and topoisomerase and gyrase when we talk about antibiotics, antimicrobial agents, and cancer chemotherapy agents. And they're just quite simply regulatory proteins that help unzip and transcribe the protein here. A transcriptase is a transcribing protein. We'll talk about transcriptase when we talk about reverse transcriptase inhibitors. And so it is a protein that transcribes. And so here a protein is transcribed one arm of the DNA to make an RNA. And that RNA can be carried out to the nucleus. Maybe this is a messenger RNA carried out from the nucleus to the ribosome. And then all that has to be wound back up again by these regulatory proteins. Or maybe we have a cell that's undergoing mitosis, maybe a stem cell, hematopoietic progenitor in our bone marrow that's rapidly reproducing. It has to undergo mitosis, so it needs identical copies of its RNA, identical copies of its DNA for it to undergo mitosis. And so all these regulatory proteins will unwrap and transcribe and then rewrap. And so these will be the things that we talk about, especially at the end of the semester when we talk about cancer and chemotherapy, antimicrobial agents. We'll talk about the stuff when we talk about HIV and AIDS and immunology as well. And so that's why we cover this stuff in the first couple of lectures. So that's cellular physiology in the most simplistic terms that I can think of. When we come back, we're going to actually start talking about general principles in pharmacology. So I did want to spend time with these lectures going over some basic principles in biochemistry, basic principles in cellular physiology, so you will have a really good foundation to build your pharmacology knowledge moving forward. So until next time, aloha.